I'm Elizabeth Blackburn. I'm a professor at the University of California, San Francisco, and I used to be at the University of California, Berkeley.
And I'm Joe Gall. I'm a staff member at the Carnegie Institution, Department of Embryology in Baltimore, where I've been for the past 22 years. So Liz, why don't we start right at the beginning. You were born in Tasmania, which is certainly halfway around the globe, if not at the end of the earth. Did you grow up there?
I did. I grew up there until I went to my last year of high school. So I grew up in mainly in a small town in Tasmania called Launceston, which had about 70,000 people in it, and I think still does. And I grew up in a large family.
You say a large family.
Yes.
Was it an academic family, or what type of background did you have?
Well, both my parents were family physicians, and I was the second of 7 children. So books and science and I think, some sort of love of knowledge was always a part of my family and always part of growing up.
What sort of things did you do as a child that might have led to your later interest in academic science?
Well, I always loved animals, and I think that really was the focus that got me into biology. Although I didn't think of it as a focus, but I was just immensely curious of animals. And one of my favorite things when I was growing up was enumerating all the different pets that we kept in our quite large garden.
So were there particular people that might have influenced you to go into science, other than your parents, presumably?
My mother's father and her grandfather were both geologists, and my father's grandfather, I found out quite late in life, actually was an avid coleopterist and had spent years in Hawaii making a very extensive collection, and he continued that activity when he moved to Australia. And we have all this correspondence where he's trying to sell his big collection to the British Museum. And that was where I first became aware that I had more scientific genes in my family than medical genes. And I remember, for example, having access to really, really wonderful books. And so people would give me books. And I had the biography of Madame Curie, which is this very adoring biography written by her daughter. And there was also another book that I just loved, and it was by Jacob Bronowski, and it was about science, and it had the most beautiful illustrations of very abstract sorts of depictions of science, and I just loved the whole aesthetic of that, and I think that really captivated me about the idea of science, even though it wasn't about biology. So the other kind of good influence was I had a very good high school biology teacher, and I had a good chemistry teacher who got me sort of excited about the world of chemistry because she was somebody who made chemistry fun. And I was at a small girls' school, and chemistry was a kind of late addition into their pantheon.
I assume it was more or less assumed that you were going to go to university. So what was the decision? How did you make the decision to go to Melbourne?
So by then my family moved to Melbourne, and I did my last year of high school in Melbourne. And in Australia, the tradition is you go to university or college almost always in the state where you are.
Liz, your description of your childhood experiences reminds me a lot of my own, where I really had almost no formal introduction to science, but I was doing science on my own in the way that you describe of having animals and experiencing and reading about it in books. So it's very interesting what parallel was going on there.
Well, I hope you weren't as wicked as me. I just remembered I discovered the wonders of touch powder. Remember this thing where you have iodine, solid iodine and ammonia, and you can make a bang? And I discovered this in high school and thought it would be very amusing to show off my chemistry and have the headmistress walk up steps on this banging stuff. And they sent the one male teacher in our school, the math master, to speak to me about this. And he said, "A little knowledge is a dangerous thing." And it was actually, they handled it very well, because, you know, it was a pretty bad thing to do, actually. But I was a good student.
So getting back to your education at Melbourne, you went on to do a Master's of Science in Biochemistry,
Yes.
I believe. Were you, at that point, already certain that you wanted to become a research scientist, or were you marking time as to what many graduate students do?
No, the Masters had a completely different significance. So what had happened was that I had decided to go to Cambridge to do my PhD, and I'd applied, and the rule was that if you didn't come from England but came from the colonies, and I don't know whether the US was honorary ex-colony for this, but if you came from the colonies, you had to have had a year of full-time research before you were allowed to come to Cambridge's august PhD program. And so to achieve that year of research, I chose to do a master's, because that was just a straightforward way of doing that. And that actually worked out very well, because my master's advisor had worked with Fred Sanger just after the Second World War. And Fred came through visiting Australia at some point, and I met him and talked with him, and then we arranged that I could go to his lab, and so it worked well.
That must have been quite a decision to leave Australia and go halfway around the world to do your graduate work.
It wasn't an unusual decision for people in Australia interested in academics, and generally the feeling was that if you could go overseas and train at some point, that was very much to your advantage.
So you were at the MRC in Cambridge, and the MRC had and still has an almost mythic reputation for producing genius. What was it like to work there in that atmosphere?
It was great fun. I loved it, because I'd left Australia, and I think the whole, you know, thing of leaving and being on your own, and it was very liberating. So I think I came very predisposed to enjoy it. And I think I sort of plunged into it fairly thoughtlessly, in the sense that I didn't stop to compare it with what other alternatives might have been going. I just, you know, was there. I'd made that decision to go, and I arrived pretty ignorant of any nucleic acid chemistry, which was what Fred Sanger was doing at the time, and that's what my research project was all about. But on the ship coming over, this was funny, I took a ship from Australia to England, because in those days the P&O line would, for free, have people who were going from Australia to Europe for free go in their first-class empty cabins.
That's the Pacific and Orient.
The Pacific and Orient line. So I spent 4 weeks, and so I'd go to the ship's library and I would read these copies of the papers that Fred had published on, and so this was my first introduction to things like phage RNA and so forth, because I'd had a very classical biochemical upbringing. So I went to Cambridge very, very ignorant, kind of a completely blank slate, I would say, in terms of molecular biology.
You said the ship's library. Did you? You must have brought the papers with you.
Yes,
you didn't find anything like that on the ship.
Did not have the volumes of Journal of Molecular Biology that any proper ship's library ought to have. No, right? No, I would sit there, because that was the quiet place where I could sit, and people wouldn't wonder why I was reading Journal of Molecular Biology reprints. But I would read those things on there. But I came kind of you know, a completely blank slate with respect to molecular biology. So I was like a sponge. I was trained very much in biochemistry. And I remember one big shock, which was that I was very quantitatively trained. And this whole idea in molecular biology that you didn't actually run standard curves, but you kind of did things in this whole other way of thinking, which was almost more digital. You know, the answers were it was or it wasn't. It was a very elegant way of thinking, where you didn't have to dirty your hands with such sort of low-class things as standard curves. And so I quickly got seduced into all of that wonderful thinking, because, you know, standard curves were very tedious things. That
I think that's what's been the definition of a molecular biologist, is a biochemist without a license. But
yes,
you had the license, so you were
I had a license,
shocked.
but I threw it away. But I willingly threw it away, because it was great fun to be there, because I think people had what I now look back on, you know, somewhat more acerbically, and look at as somewhat of a sense of their own importance. I mean, people, it was very self-referential. Science went on, and it was really how it related to what was going on at the MRC, and whatever was done was always compared with that. But it was fun for somebody young to be in that kind of heady environment where, you know, you talked with Francis Crick, and Fred Sanger was my advisor, and there was a complete informality about it. And when I started to refer to Fred as Dr. Sanger, everyone just said, "You mean Fred?" And so there was this whole culture that you didn't have hierarchies. And they didn't really know what to do with students. That was the wonderful thing, because there were very few of us, and most of us came from overseas, and there were very few of us. So we were in the Cambridge biology PhD program, and our advisors were advisors of various scientists at the MRC. So Fred got £8 or guineas a term for supervising each of us students, and he always gave it back to the university.
You said you had fun,
Yes.
and obviously the way you're describing it, you enjoyed the
Yes.
experience there, but a lot has been said about the difficulties that women have had in the British educational system, particularly the British university system, and
Yes.
even still do.
Yes.
You don't seem to have been affected by that, or were you affected by that, or
I was certainly...
were you allowed to take tea with the rest of the men?
Yes. So the atmosphere in terms of, you know, being in the lab and being a member of the research community there was to my mind just fine. You know, all the conversations I was in on and joined in on. However, part of me was very aware that long term, to be a scientist in Britain would be something very, very hard to break into, and I think particularly as a woman. And so my eyes just turned to the US. You know, this was in the mid-70s and early 70s and mid-70s, and so it was very clear to me that this was never a place that I would think about going into long-term.
So they were willing to educate
Yes.
you, but not to let you into the club? Is that
Well,
what
I
you're
never
saying?
asked them if they were
I
willing
see.
to let me into the club. I think I made
Right.
some assumptions. You know, I looked around, and it was fairly clear that women scientists were rare, and they had, you know, in some way accommodated in some way. But I think in my heart I knew I wasn't going to be comfortable with that.
Now, as far as your thesis project itself is concerned, did you choose that? Did Fred Sanger choose that? Or did you discuss it with him and come to some mutual understanding? How did that happen?
Well, it was definitely, Fred said, you know, why don't you do such and such, which meant do this. But basically, there were these little fragments of phage DNA which were out in the lab, and people were trying to figure out ways of sequencing them both directly and, in my case, indirectly. And Fred said, why don't you try copying them into RNA? Because people knew how to sequence RNA, because he'd worked out methods for sequencing RNA by patching together little overlapping fragments.
Oh, I know all about that,
Yes.
because I
You did it.
I did it.
I
Yes.
know. And so that was the project that Fred chose for me. And then how I went about it was very much left up to me, because that was the sort of laissez-faire kind of way things were done.
But your project was actually sequencing some 40-some bases of,
well,
wasn't it? Something
I had
like
two.
that?
One of them was a 48 nucleotide sequence, and then to our great pride, we have another one where I think it was 130 something. There was a second one as well. And it's so funny to think that this was actually exciting at the time, to get this little patch of sequence. And, you know, it was a DNA sequence, and nobody had ever had actually a DNA sequence that long. But we didn't know, you know, that the DNA and the RNA and the protein would all align. And that was actually quite exciting, and somebody else provided information for one of the segments of the phage DNA, that the protein aligned with the code that you could read directly in the DNA. And in some ways it seems obvious. In other ways, you know, you now think, whoa, what if there'd been a splice or something in there, right? So it is fascinating in hindsight to think about what at the time sort of seemed, oh good, that's confirming the linearity of the genetic code. But it wasn't totally obvious that it was going to be like that.
And how long did it take you to do this?
It was just under 3 years, but there was no coursework. And again, you know, when I wanted to take a course and I looked around in Cambridge, there was very little on offer in terms of coursework that wasn't undergraduate coursework. And so most of my graduate education was through seminars and osmosis in terms of the PhD part of learning molecular biology.
So when it came to publish this work, you published
Yes.
it in the Journal of Molecular Biology,
Yes.
I believe, which was
Yes.
the most prestigious
That's right.
journal at that time.
That's right. Almost the house journal, I would say, because it was, one that was very much identified with the molecular biology community in Cambridge. Fred was working on at the time. When I was doing the RNA part, he was working out what became, of course, the ultimate method for sequencing DNA, and he was doing that himself. And so that, would be published in a more high-profile journal.
But it must have been true that part of the education, in a way, is introducing you automatically to the best in everything, so that you come to expect it in your work. That is a very important aspect of that kind of education.
Yes, I think that's so important in a training environment, that people get a sense of how you do good science and what it means to do good science, and have a lot of very critical discussion about it, because there were these sort of famed morning and afternoon teas that the British do, and they were actually very productive times, because people would sit around and just talk science endlessly, which I just loved, and at lunchtime too, because the lab was a couple of miles out of Cambridge, and so there was pretty slim pickings in terms of food. So basically it was much better to eat at this cafeteria, which Max Peruzza's wife ran out of the goodness of her heart.
Liz, you mentioned that when you finished sequencing a bit of the Phi-X molecule, that you could see the sequence predicted the amino acid sequence of the protein. That reminds me that at just about the same time I was using the same method, actually, of sequencing DNA by sequencing the RNA made from it. And we were working on a very simple satellite-type DNA from Drosophila. And I remember very clearly that we sequenced both strands. And of course the sequences were complementary, and I didn't think anything of it. I don't think we even mentioned it in the paper. But thinking back on it, I think it may have actually been the first direct proof that the two strands of the DNA are complementary by sequencing.
And how many assumptions there are that we just take for granted that are actually not necessarily proven rigorously and experimentally.
How did you make the decision to come to my laboratory?
Well, I had met my to-be husband, and so at sometime late in '74 we decided to get married, and John had a temporary soft money position at Yale, so I immediately thought of your lab and had been very impressed by the fact that you had gone straight into DNA sequencing, which is something I loved, and came at it from your biological background and perspective.
Well, at that time in New Haven, as you well know, we were working on the protozoan Tetrahymena, and we had discovered that its gene coding for ribosomal RNA was an extrachromosomal molecule, something like an episome. And when you came, we began to make a molecular analysis of that molecule. And when you actually entered the laboratory, we decided that you were going to sequence the ends of the molecule. Do you remember how that decision was made?
Well, I remember from my side at least, it was very dictated by the one thing you could do in these pre-restriction enzyme, pre-cloning pieces of DNA days was people could get at the ends, because there was a wonderful precedent in lambda DNA that people had actually managed by very tedious methods to get at the sequence of the overhang of lambda linear DNA. And so I thought, well, wouldn't this be wonderful to look at a real nuclear linear DNA, a real one from a eukaryote, as opposed to merely a virus. And so I was very impressed, because you had these marvelous linear molecules, and some of which, you remember from the electron microscopy, were circular. You saw occasional circular ones. And so to me that just looked like lambda, which of course was wrong. But I do remember the discussion we had, though, about should it be oxytricha, which David Prescott had shown had very short, heterogeneous population of short molecules in its somatic nucleus, or should it be tetrahymena? And I remember you said, it's such a, not in those words, but it's such a pain to grow oxytricha.
Oh, indeed.
And tetrahymena, here it is, this basically pure species of molecule that you had worked out how to purify away from all of the rest of the DNA. And so that was a very compelling argument to me that this was, you know, clearly the molecule to work on.
So at the time, I think probably both of us thought that sequencing the ends of a molecule was a somewhat esoteric exercise. As they say, for the sake of our TV or radio audience, what was the larger significance of sequencing these ends?
Well, I'm actually going to come back slightly to what you were saying earlier, which was, well what was the significance? And I think I had grown up with, or learned from this molecular biology that I'd been imbibing, that things in molecular biology worked in generalizations, and if you found a solution, there would be this elegant uniformity throughout life, and, you know, like the genetic code worked throughout life. If you're a, you know, a beetle or a bacterium or a, you know, a baboon, I mean, it would all be general. And so I think I had this underlying touching faith that if you found something in a nucleus, it would be general, because surely nature would be elegant. Now, you know, we lucked out, because these little molecules, which we didn't, as you say, have really any reason to think would be typical, it turned out they were typical, at the ends at least, of the much larger chromosomal linear DNAs that are found, you know, in all eukaryotes pretty much generally.
Right, but the truth of the matter is that at the time you did the experiments, and I think for at least a year afterwards, we didn't know that the sequence that you had determined for the ends of the tetrahymena molecule were similar to sequences in any other organism or organisms in general. When did you actually first come to that realization?
Well, there were two steps. Remember, with Meng Chao Yao, who was a postdoctoral fellow in the lab with me in your lab at the same time, and we did an experiment where we tried to label up other portions of the genome of the somatic nucleus of tetrahymena, and we found that this same sequence, which we always referred to by its C-rich strand, the C4A2 repeat strand, that C4A2 repeats were present in multiple other locations in the macronuclear genome.
Right, but that was still just tetrahymena.
Definitely within tetrahymena. Yeah. And so, and then again, it was I think the world regarded tetrahymena as very weird. You know, the nuclear dichotomy. Still regarded as weird. And I never thought of it as weird in the sense, as I say, I think I did have this touching faith about molecular biology, you know, at bottom would be fundamentally conserved.
Eventually, of course, it was found that this sequence or something extremely similar to it was at the ends of all chromosomes.
Yes, so I think you're absolutely
except
right.
for Drosophila.
Yes, right, with huge irony, since the telomere was named by Muller in Drosophila, right?
Yes. So, well, shortly after leaving my lab, you took a position at Berkeley. I think you did a very short stay at UCSF and then went to Berkeley.
Yes.
So what was it like to finally have your own laboratory?
Well, I loved it. I just felt like I had a playground, in the sense that I really felt tremendous freedom to do exactly the research that I wanted to do, which is something that I think it's harder for people now. I think they get lots of advice, lots of it very well-meaning advice. And, you know, anybody giving me well-meaning advice, you know, would have told me to do far more sensible things than work on this crazy organism. Did
you start right away working on the enzymology of the ends of the molecule? In other words, looking for the enzymes that added the telomeres to the chromosomes?
No, the chronology was that just before I left your lab, I was looking at what was the chromatin structure of the telomeres. And I remember I have a southern blot, which was, I think, the very last thing I did before I left your lab, which was showing a non-nucleosomal protection of the C4A2 repeats, as we call them, from the nuclei of tetrahymena. So it was essentially looking, most of those molecules were the amplified ribosomal RNA genes. So I went thinking very naively that I would find the proteins that were protecting those ends, little realizing what a biochemical bear that was going to be, and I never succeeded. And in fact, it took yeast genetics to give people an entree into that problem. So I was fascinated by the proteins that were associated with the telomeric DNA and fascinated by the structure itself, which had complexities, if you remember, that to this day are still not addressed.
Right.
The single-strand discontinuities on the C-ridge strand are such a mystery and still a technical nightmare.
And the nicks.
And the nicks, as we called them, but they were actually single-strand discontinuities because they were non-ligatable.
Yes.
And so, I mean, single-strand discontinuities by a sort of process of patching together information. So that's what fascinated me, and it was only in the very, I guess around 83, I started putting together thoughts from various experiments that made me think that there might be an activity that could make telomeric DNA. And that was evidence coming from tetrahymena experiments, looking at the rearrangement of the chromosomes, in which one sometimes gets telomeres added de novo to sequences where there weren't any before. And that was emerging in our lab. And Prescott had seen something similar in Oxytricha right around the same time, like 82, I think we both found that kind of thing. And then work in yeast done with Jack Shostak, where we'd put telomeric ends from tetrahymena into yeast and found that telomeric DNA, which we deduced it was getting somehow stuck onto those ends. And those were the kinds of experimental lines of evidence that made me think that there'd be an enzyme. But it wasn't right away.
But after you did decide that there had to be an enzyme or some activity that added ends to the molecules, did you develop a full-fledged model that you then set out to prove, or did you, maybe stumble is not quite the right word, but did you move gradually and slowly to the realization that there was an enzymatic activity?
Well, I think there was two parts. One was thinking about how could you explain, two things. Why did telomeric DNA sequences get stuck on other sequences that didn't look as if it was through recombination? And remember, the reigning hypothesis was how you join two DNA sequences together was through homologous recombination. And the yeast genetics had amply shown that. And so this just wasn't fitting that. And even when a telomeric tract of DNA got progressively longer, you'd have to invoke some very odd kind of recombination that somehow added, unequal crossing overs and more steps, so that you got this sort of steady lengthening that had been seen in certain settings. And so it sort of wasn't adding up. So the idea, well, is there an enzyme? And I actually remember I gave a talk, I think it might have been UCSF, and some of us were talking after the lecture, and I was saying, maybe something adds to the end, sort of throwing it out. And somebody said, yeah, that could explain it, and it was real reinforcement.
So when did you first do an experiment where you saw this progressive addition from templates?
Right. So that was somewhat gradualistic, because so here was this idea, and I thought, well, there's one way one can find evidence for such a thing, and that is actually to do biochemical experiments, because I was realizing that yeast genetics was beautiful, but it was just not answering the questions, because it asked the questions and one was answering it sort of in the same terms. So I realized that one had to, roll up your sleeves and just try and do a biochemical kind of analysis. And so the very first thing that I thought would give some hope about this was actually done for a completely different reason by my student Peter Challoner. And Peter was trying to look at why the tetrahymena ribosomal RNA gene molecules are palindromic. And so you had discovered, with this lovely work with Kathy Kara at the same time that I was a postdoc, that there was a palindromic arrangement of the sequences in the rDNA. And Ming-Che Yao had found that there was a single germline copy that gives rise to that. And so how did it turn into a palindrome? And so somehow palindromes get generated from that single molecule and then amplified up in some order that wasn't necessarily clear. And so I was one of the people fascinated by that question, and so Peter had devised an assay in which he made very small linear sort of versions of what we thought might be a pre-palindromic molecule, very shortened version of the linear ribosomal RNA gene, and it had, you know, a telomeric end at one end, the normal telomeric end. And so the bottom line was that he incubated these molecules in a mixture of reagents plus an extract from tetrahymena cells at the time when those palindromes get generated at the biological stage in the development of tetrahymena when they're generated, and one can make extracts from synchronized cultures of cells. And so he incubated these substrates, and he didn't see any palindromes being made, but he did notice that the restriction fragment that contained the telomeric repeats started fuzzing up and moving up in the gel. And I had been thinking very much about this question of, you know, what might be accounting for why sequences were appearing on the ends of DNAs that didn't formerly have telomeric DNAs, or why a tetrahymena telomeric DNA sequence in yeast got telomeric DNA added to it, or why, in fact, telomeres get longer and longer, as they had been shown to do in trypanosomes. So all of these things were pointing toward the idea something might be adding DNA to the ends of telomeres. And so seeing Peter's result was not only exciting because you could see the band moving or sort of fuzzing up a little bit in a way that suggested the possibility of something being added, but also just the idea that it would be feasible because these extracts were not turning the DNA into, you know, rubble, into mononucleotides, which was a very real fear if you did an incubation in such a crude extract as we had chosen to use. That was one of his very last experiments, and he wrote his thesis up fairly soon after that, as I recall.
So you said that you saw the bands fuzzing up.
Yes.
But you also implied that you immediately realized that this could be adding sequences. When we get a fuzzy gel,
yes,
we often just think, well, we did it wrong.
Yes, well, this had a nice sharp band in exactly the same lane, which wasn't showing that effect, you see. So it wasn't a technical issue of how the gel was run. And the fuzziness appeared in a time-dependent fashion for that band, but not for the other bands on the gel. So this was clearly some sort of reaction happening in a time-dependent fashion, which certainly didn't say that there were repeats being added. So, but that encouraged me to myself, and at this point I thought this was pretty far out, so I didn't want to bother any student with it at that point, but at that point I started incubating restriction fragments with this extract, with and without telomeric repeats on the end. And the thing that really encouraged me was that I took some unlabeled restriction fragments and put them into this gamish with the extract and, you know, everything I could think of, triphosphates, ribotriphosphates, ATP regenerating source, you know, everything I could think of that might make anything replication connected happy. And then I probed the products with the C4A2 repeat oligonucleotide probe, a labeled probe of a southern blot of those reaction products, and bands appeared that were fuzzy and got more strong signal over time. So I thought, aha, there is something in there that makes C4A2 repeats appear in places where it wasn't before in a time-dependent fashion. And I liked the fact that, you know, the 30-minute and the 90-minute time points, you know, they look different over time.
Right.
That was good, right? And then I was joined in the lab by Carol Greider, who came as a graduate student starting at the Berkeley program.
But that's a story I want to get into.
Yes,
but
yes.
Before we do, I just wanted to ask you, you said that you took the project over when
Yes.
you saw that it was
Yes.
getting to be chancy.
So,
I was just wondering if you felt uneasy giving a student a problem
Yes.
or letting the student...
Definitely.
Because that's interesting, because in a way the best students are the ones that take a problem that
Yes.
doesn't necessarily have a solution
Absolutely.
and work it out.
Absolutely. And I had just got tenure, and I was feeling very brave. And
Tenure system does have its advantages, doesn't it?
well, I didn't like the process up to it, but after that, I was feeling, hey, I can do what I like, and I wanted to do these incubations. But then I thought, well, this is fine for me to do some experiments myself, but I'm not going to be able to be doing this all the time. I was by then, an associate professor, and they start bringing in the committees
Soon
and
to
the
be
teaching,
chair, yes.
And the chair took a long time, but a good 10 years before I was ever a chair. But at that point, Carol joined the lab, and I said, well, I have this, a list of projects, and here's this crazy one. And she said, great, I'm interested in the crazy one. So I thought, aha, this is wonderful. There's somebody who was willing to take on a risky but interesting, novel sort of project.
And she's a graduate student.
She was a graduate student.
Yes.
So Carol's job then was to home in and get a proper assay where you could really say what was being added to, because at this stage I didn't know whether it was being added to, a tiny bit of telomeric sequence, or whether it was being added to a completely non-telomeric sequence.
So Carol's first job then was to define the sequences that were being added to.
And so she started in with restriction fragments where she would, have a telomeric sequence at one end and a non-telomeric at the other end, and then try to make the restriction fragment ends, have 5' overhangs or 3' overhangs. We didn't know what would work properly.
Right.
And then the breakthrough was to sort of say, aha, and I think we really both thought of this at the same time, why not use a DNA oligonucleotide just to get the concentration up? And it's very banal now, but DNA oligonucleotides were kind of exciting new things that you could get synthesized in those days. And
I think it's always difficult in tracing the history of these ideas, yes, to realize how much is dependent upon technology and how much is dependent upon what's available, because
yes,
nowadays
yes,
a student reading these papers would say, well, that's obvious. I mean,
yes,
we get oligonucleotides
yes,
daily.
Yes,
but
yes, but this was now something that you could actually get somebody to make you a synthetic
yes,
DNA oligonucleotide. It was, how exciting. We could add lots of it and just
high,
have more of it, because we could see these little hints, and we just
right,
knew we weren't getting enough signal, because by then she was trying to look directly at the radioactivity being added to the restriction fragments. In my initial experiments, I had done it unlabeled, so that had given me lots of high concentrations of triphosphates, I think. And now I came in with a radioactive probe and could see the sequence getting added. And then the other thing was Carol was then able to now resolve the fragment products much better. Once you start with a short DNA oligonucleotide, now you can run sequencing gels as opposed to the much more crudely separating gels that we had to use with restriction.
So now your smear
yes,
was turned into bands.
Well, there was just this wonderful first gel that
yeah,
she ran, where now instead of being a smear, there was a periodic
right,
set of bands, and I think we'd run enough sequencing gels to know that they weren't separated by an interval of one base.
But 6.
And so, of course, 6 was the number that sprang to mind, but it wasn't obvious from the very first gel
yes,
that it was 6, but kind of knew it had to be 6, I guess.
Right. No, so by that time, then you had basically your assay. That was the
assay, yes,
critical thing, was
yes,
to get an assay.
That's right.
Then
yes,
Carol and you went on to do what is undoubtedly one of the most important experiments in molecular biology, which was to prove that the addition was being carried out by a ribonucleoprotein.
Yes.
So
yes,
lead us through the
yes,
steps in that development, because that certainly is a fantastic discovery.
yes. Well, the very first thing that we thought of was, okay, we've taken a crude extract from a cell that we know contains tracts of C4A2 repeat, G4T2 repeat, telomeric DNA. We know that synthesis templates off DNA. So the very first thing that we wanted to ask ourselves was this just copying some contaminating DNA that was in these crude extracts?
These things which are obvious possibilities, they get forgotten when you read
yes.
the final
oh yes,
paper.
Yes. But that was the obvious thing
Yeah.
that just jumped out, and so there were a couple of ways to do that. And one was, of course, to purify things further, but the quick way was to try to get rid of the putative templating DNA and use a nuclease. And so we thought, well, we want a nuclease that will allow us to come in and assay with a primer again, i.e. a DNA primer. So we thought, whoa, micrococcal nuclease, perfect, because it's got a calcium dependence, and so we can get rid of it, and then we can now let our reaction, which by then, I think by then we'd shown required magnesium, but I think we just assumed it would require magnesium because polymerases did, and I can't remember if we tested the magnesium dependence at that stage. Anyway, so the idea was we would just get rid of the contaminating putative template with micrococcal nuclease, and that was a most fortuitous choice because micrococcal nuclease actually destroys RNA as well as DNA. Now, we did it in a very sneaky way in retrospect, but we didn't know we were being very lucky. So we used a very crude extract and got the micrococcal nuclease to do its thing on that very, very crude extract, which had a lot of contaminating DNA. And we could see the contaminating DNA was getting broken down into very small pieces by the micrococcal nuclease, but the ladder of product bands that was produced didn't get any shorter. So that was good. And then in the meantime, Carol was starting to biochemically purify it to start to do the first steps of cleaning up the extract. And so on a much, much purer extract, we then quickly noticed that now this micrococcal nuclease actually destroyed the activity.
Right.
So it was that lucky window of if we destroyed the activity right away, we would have thought, oh well, it's just a contaminating, DNA that's templating this reaction, and probably, who knows, might have not gone on with it. But luckily, the experiment was done in 2 stages. This is where quick and dirty paid, because the crude extract, there was enough DNA to sort of use up all the micrococcal nuclease and keep it busy. Probably there was tons of RNA too there, and it didn't trash all of our enzymes. RNA component.
But now, you're speaking here of using micrococcal nuclease and the
Yes.
magnesium dependence
Yes.
and so forth, and all this sounds highly reminiscent of the kind of thing that Tom Cech had been doing, I think, previously, showing the self-splicing.
Yes.
So were you thinking along those lines?
We weren't thinking along those lines. We certainly weren't thinking that there would be an RNA component in there at all.
This was not a preconceived idea that you were trying to prove.
No, far from it. I mean, it was a serendipitous observation which had come out of our trying to ask whether this ladder of products we saw was just being generated because there was just copying of random fragments of C4A2 repeat containing DNA. And the reason you would have seen them was, at this point we just had CNA in the radioactive triphosphate mix. And so that sort of sequence we know by just random nick translation would get labeled up from the contaminating genomic DNA. But Tom had by then got very nice sorts of conditions for what you put in extracts. And so we definitely used the concentrations of triphosphates and things that Tom had used, because those had been worked out for nuclear reactions in crude nuclear preps that he'd been using in terms of the splicing. But RNA didn't cross our minds until it got that micrococcal result. And there was a great reagent you could buy from Sigma, which I don't think you can buy now, which was a ribonuclease A, which has the one of the residues in the catalytic active site is oxidized. It's chemically oxidized. And so it's a dead form of ribonuclease. And we were very aware. I mean, I'd grown up on this idea. Ribonuclease is this little basic protein that can mess things up. And so here was this marvelous control. And so Carol immediately did that control. And lo and behold, the ribonuclease with the dead catalytic site didn't destroy the activity.
So you knew it was catalytic activity and not just
this basic protein glomming
right,
onto
right,
nucleic
right.
acids and unspecifically inhibiting the reaction.
Now, I remember very well the paper in which you and Carol described fishing out the right RNA.
Yes.
And what I remember about that
Yes.
paper was that there were just an enormous number of RNA bands. How did you pick the right one? How did you?
It was just it was brute force. I mean, Carol really just did a terrific job of just going in like that, you know, like a, I don't know what you just, you know, she was going to get that RNA out. And it took, I think it was 6 columns, and it was basically the most classic biochemical thing, which is that you run some sort of gradient, you assay for activity across your fractions, and you look and see what RNA species goes along with it. And it was down to tRNA-sized stuff, and this wonderful little band. There was another one for a while, but that
Which you
went
could
away.
see on the gel.
And you could see it by radio labeling up all of the RNAs in that fraction with something that radioactively labels the end. So there was the most, you know, these hideous gels, right, with all these other contaminating RNAs. But the one that came, you know, along, and as I say, it was down to tRNAs and this one. And I forget why we eliminated tRNAs. Actually, there was some reason why we eliminated them. And
seemed pretty unlikely.
And then this little band, which you could cut out from a gel with big scale-ups, could be got out in just enough amount for Carol to get some partial RNA sequencing, you know, bits of oligonucleotide patches of sequence.
So that was done by the classical RNA sequencing technique?
Yes, and because it was such a low amount, it was only possible to get just little patches. But now, and that enabled Carol then to go in and fish a gene out.
Now, there's actually, in this story, as in so many scientific stories, there's the part that you don't tell.
Yes.
And it seems to me one of the things here that you don't tell is that if you hadn't done this on tetrahymena, in fact,
Yes.
on conjugating tetrahymena, in fact, at exactly the right stage, you wouldn't
Yes.
have found telomerase at all, because telomerase isn't just everywhere.
Well, it is everywhere, but it's in
Well,
meager amounts.
that's what
yes, so
I mean.
tetrahymena was this incredibly good choice. And you know, Joe, you take credit for this, because I internalized from you, choose your biological system for your question. And so the multitude of telomeres in tetrahymena and its biochemical sort of friendliness, you know, you could grow up big flasks and get lots of it. So those lent itself to tetrahymena. And then the biology, you know, handed itself to us on a plate, which was this time in the development when it amplifies up the ribosomal RNA genes and it is fragmenting the genome, and it's busy in the business of making telomeric DNA at this window of time in its development. And so that certainly, that gave itself to us as the time when you could most likely get it. Actually, we got, in the end, we used to just get it from vegetative cells, but it was the highest concentration.
I was going to say that you tell the story as if it was obvious that you should use the conjugating cells because
Yes.
because of this, all of which is
Yes.
the biology.
Yes.
And yet,
yes.
had you done it with vegetative cells, which probably the average biochemist would have done, you might not have found it, right?
We certainly found the activity with the oligo primers, the oligonucleotide primers. You could see it in vegetative cells right off the bat, but it was less. Now, whether this initial experiment where I was using C4A2 repeat hybridization to get new bands that lit up over time, whether that would have worked in a vegetative extract, I don't know if I ever tested that. But definitely the conjugating cells, that is the cells going through the developmental stage where they're making new macronuclei and making telomeric DNA, you know, that was crying out to be used, because that was at least a chance where you thought there would be some action going on.
Right. So at least there is a message here, is that you want to know your material and to use the biological material that's most suited for the particular question.
Yes, yes. My sense is always make those cells pay for their keep. Right?
So
we house them, we feed them, make them work.
right. So In subsequent years, a lot has been made about the role of telomeres in both aging and in cancer. I know we could discuss these topics for several hours, but just broadly speaking, what has been the significance in those two areas of the work on telomeres, in aging and
Yes.
in cancer?
Well, actually, I've undergone quite an evolution in my thinking about what telomeres and telomerase may be doing in aging. And so the initial idea that was put forth was that you got born, your telomerase was turned off, your telomeres ran down, your clock ran down, and you died. And that
Depressing
is actually
story.
a depressing story and experimentally absolutely true for yeast cells. So, and in fact, when we mucked up telomerase inadvertently at first in tetrahymena, that's sort of what it did. It went on for a while, and then the telomeres were getting shorter, and it actually stopped.
Right.
So anyway, that sounded sort of too good to be true, to be honest. So laboratory strains of mice have very long telomeres, and yet they die at 2 years of age, pretty much, of old age, and their telomeres are certainly much longer than ours are. Although, you know, there's a general trend downward in human telomere length with age. So I thought this was really pretty shaky, and I wasn't really buying it. But I've become more intrigued by observations about what goes on in aging. An inadvertent experiment with people who come to the clinic did first emerge in 2001, which people who are essentially haplo-insufficient for telomerase RNA die of progressive bone marrow failure, and so they never make it past middle age, and in fact they can die even as young adults. And the cause is that they exhaust their immune system. Nobody knows if it's the stem cell compartment or what that gets exhausted. So that set the stage for the idea that at least telomerase, in this extreme situation of only a 50% gene dosage, can be limiting over, you know, the decades of a human adult life.
Were the telomere lengths actually measured in any of those patients?
Yes, and they have very short telomeres.
They do. Is that, excuse me, is that a mutation in the RNA gene?
Yes, yes. Talk about, you know, bad luck. This is 451 nucleotides long. I mean, there's a tiny target.
Right.
And so other studies, though, have been done in which people have linked mortality rates to telomere length. And I think a very interesting one was done by Richard Cawthorne and published a couple of years ago, 2003, in which these elderly people who were all older than 60 died of various causes. And what they found was that those with the shorter telomeres had much higher mortality rates overall, and they had higher mortality rates specifically from cardiac disease, which is one of the major killers, as well as from infections. So that was interesting given that immune system compromising that happens when telomerase RNA is haploinsufficient. So those are intriguing results, and we've looked actually very recently in peripheral blood mononucleocytes, which is pretty much the only cells a healthy person will give you, and assayed for telomerase in this quite heterogeneous cell population, and we found telomerase activity has a range of 15-fold among individuals. Now the startling thing was that the high and the low telomerase people were different with respect to 6 of the major cardiovascular disease risk factors at numbers that achieve, you know, statistical significance. So all of this is adding up to, I think telomerase might be important in, and telomeres might be important in aging from these very common causes of mortality, which are the ones that frankly get most of us. And if we're lucky enough to, you know, pass all of that, then I think there's a lot more programmed genetic pathways that kick in and become apparent at that point.
Okay,
So,
but now as basic scientists, correlations are nice.
yes.
But how can you go mechanistically from a
Yes.
telomere to a heart attack?
Yes, indeed. And so people have looked, for example, at telomere lengths in diseased arteries, and again they find that they're much shorter. So one idea is that as the arteries become diseased and they're trying to replenish themselves, they're, you know, over-replicating, if you will, and maybe exhausting their ability to replenish themselves. That's one possibility.
Right.
The clinic is a really terrific assay system because it's so sensitive and can be very well defined. So now you try and work back and you say, you know, is your favorite gene underlying the whole clinical pathology? Well, you know, we can imagine that that might be true in a few cases, but
or is it
the
just
question is,
aneuploidy?
how does it work?
Is it aneuploidy?
That's the question. Or is it low telomerase? Because we know telomerase now seems to be prompting cells to have different gene expression patterns compared with if it's up or whether it's down, well before the cells show any sign of exhausting their ability to keep multiplying.
You mean telomerase itself
Yes.
has an effect on transcription
yes.
at other loci?
Yes.
So you did this with a microarray?
You do microarray experiments and you look and say what genes
Right.
showed more or less activity after about 4 days. Now this is a time when these cells have plenty of telomeric DNA,
Right.
at least in bulk. And we didn't see any signs that the telomeres were becoming unprotected, at least by the kinds of assays that are available. So the simplest interpretation so far is that perhaps telomerase is influencing pathways that in turn end up changing gene expression profiles. So this is intriguing. Is it telomerase interacting with the telomere that's important for this effect, or is it telomerase floating free? And we don't know that yet.
So I wanted to ask you, frequently the best strategy, or certainly a strategy with problems like this, is to go to a model organism.
Yes.
Now, obviously you can do a certain amount with mice,
Yes.
but they have a fairly long generation time. Unfortunately, Drosophila is out because it does its telomeres differently.
Yes.
Does it even have a gene for telomerase?
Not that anybody has ever
Nothing.
annotated in
Okay.
the Drosophila genome.
How about C. elegans? Does it?
Yes, it does. It has telomerase, and I think that the model systems are incredibly powerful for questions of, you know, how do cells work, how do organs work. But I think in terms of trajectories of life, you know, these things that take decades in us, I think we're the model system. And so I think
you're going to have to struggle with it.
I think one has to look at that. And then one has to say, well, how is telomere maintenance, if this is the model of the day, and it's a model that I think is worth exploring, how is that being influenced by things that are not genetic? And the obvious ones, diet and so forth. And we certainly know there are connections there. And I think it's worth exploring. So I've come from a deep skeptic about this to thinking there's terrific phenotype analysis possible in people. And now it's more and more reasonable to think about actually asking about the human, not to neglect the mouse or the C. elegans or the yeast models because they're so powerful, but they frankly won't tell you
about aging.
About aging in people who live 80 years.
We started this segment of our conversation by my asking what you thought the relationship of telomeres to aging and cancer.
Yes.
And we've sort of been concentrating on aging.
Yes.
But to get to the cancer question, do you think that those are simply two sides of the same question, or do you think telomere effects on cancer are different from telomere effects on aging?
So if you go to sort of the end, the most terminal phenotype, if you will, of cancer, which is the metastatic advanced malignant cancers, and you look at them in people, there's a very striking finding that telomerase is really up in the vast majority of those. There are alternative pathways used in some of the mesenchymally derived rarer cancers, this alternative mechanism of maintaining telomeres, which, by the way, does appear to involve recombination. So this mechanism was found first by Vicky Lundblad, and you can see evidence for active recombination once you take telomerase away in the telomeres and their subtelomeric regions. Actually, it's very interesting. If you look at these yeast, again, by microarray analysis, my postdoc Shivani Natyal and Joe DeRisi showed a few years ago that these yeast that are growing quite well, apparently, on the plates by this recombination mechanism, if you do their gene expression profiling, they have the hallmarks of environmental stress response.
Stress response.
This is rather amusing. But the common cancers, you don't see that. And so that again starts raising a question, well, is it just to maintain telomeres, or is telomerase doing something else? Because why do so many cells have this really upregulated telomerase? And they have very short telomeres. They don't need a ton of telomerase that they have. And so one would say, well, there's been some sort of selection in the cancer for the cells with the higher levels of telomerase. And we don't understand that, but it smacks of there being some other role.
Well, these are broad questions which we could obviously continue to discuss at great length, but let's move on a little bit now, and let me ask you, what particular things are you interested in now in your laboratory? What things are going on, and what do you see yourself doing in the next few years?
A lot, I hope. Every review on telomeres will show some sort of series of blobs. And we actually have very little idea of what those blobs, which are the telomeric proteins, are actually doing.
So how many proteins altogether do you think constitute the telomere in vivo?
There's a probably fairly, a handful, a half dozen or so of kind of card-carrying structural ones that are on telomeres. And then there are the visitors. And the visitors are fascinating because they include the DNA damage response proteins. And those are the ones that classically people thought should never, ever be on telomeres.
And the silencing proteins, too.
And the silencing proteins accreted on to even those core set of the most immediate telomere binding proteins. And so that basic structure is just not understood yet. It's sort of embarrassing after all these years. So that's one of the very basic things. And the other is, well, how does telomerase work? Well, we sort of know and we don't know. Telomerase tantalizes us. It's a dimeric protein. And we found that if you had one RNA with a 3-base substitution in it, it was dead as a doornail, it was as good as a null. And if you crossed in with a wild type, if they're co-expressed in the same cell, then now this mutant telomerase is now utilized. And we have yet to get to the molecular bottom of that.
So when the telomerase is extending the end of the molecule,
yes,
does it stay on and just keep adding, or does it come off and come back on?
Okay. Well, we showed genetically in tetrahymena a result that I think is compelling, that it doesn't make these lovely long ladders of repeats, and the average addition was probably 1 or 2 at most. So that said that it must be very non-processive in terms of it's adding very few, at least on one RNA template. Now we don't know whether it switches to the other RNA template in the dimer and then preferentially uses that. So this business of adding long repeats is a kind of a hallmark of a healthy telomerase, but frankly the evidence that it does that in vivo is non-existent. All the evidence, it says that it adds in rather meager amounts. In fact, it's the world's laziest polymerase, is what I like to call it. It's the world's worst nucleic acid polymerase.
Liz, looking at your CV, I'm struck by the extraordinary number of things you do outside the laboratory, like NIH committees and various society activities. So what motivates you to do so much of this kind of service to the community?
I think that is such an important part of being a scientist, because A, you find out really what science is going on, and B, it's a whole other level of, it's a support as well. You talk with people and have a kind of community that you don't have in the same way in your institution, because the dynamics are different in the institution. I suppose I do find it interesting, the larger picture now.
Modern biology raises a number of other ethical issues, or a number of ethical issues, such as, say, stem cell research and that sort of thing. And, of course, in 2002, you were invited to join the President's Council on Bioethics.
Yes.
And as you well know, in 2004, you were abruptly terminated from that council.
I was invited not to stay on it.
Could you explain a little bit about those happenings?
The person who became chair of that, Leon Cass, asked me on the phone, would I do this? And I said yes, somewhat to my surprise, but it was at a very historical moment in terms of it being very soon after September 11, 2001. And I don't know if you remember how people felt then, but it was this really interesting thing where when I started forming this response in my mouth, which was, I can't do that, I'm too busy, which was the thing I would have said, you couldn't say that. And it was just, it just didn't feel right. So I said, well, okay, I would allow myself to be nominated. And then several weeks went by, because they have to go through a whole process of vetting and so forth. And I was actually asked who I voted for, and I could say, nobody, I'm still not a citizen.
In fact, I really wonder, since we have a secret ballot, is it even legal to ask someone?
Fascinating question. And the alarm bells had all gone off at this point, but I just sort of thought, hey, somebody has to do this, and at least I'm a scientist who's not vested in stem cell research, but I know some cell biology, and I kind of know my way around garbage. I think that was the thing, and I can talk to people who know, I can say, hey, is this really on the level, or is this, just a high-flown claim? And I was also
Were there
intrigued.
other scientists on the committee?
Yes, there are a few people who have some scientific/medical backgrounds, but I think I was really the only person who really knew sort of cell biology in its fairly modern incarnations, I'd say. So I started on this council and noticed that there was a bias sort of in the way we even approached things. And certainly the agenda surprised me, because I thought that the range of things was very much narrower than I'd thought it would be.
What was this range?
The ostensible immediate mandate was to examine the question of cloning, a totally valid question to be asking, and the catchword being meaning human cloning, and that right away confusing reproductive cloning from cloning, to make embryonic stem cell lines, and all of the arguments that arose from various sectors about that. And then there was issues of, should there be uses of medicine for enhancement of people, and what should one do about patenting, with somewhat of a bias towards, if we intervene by using modern medicine, is this somehow a bad idea and makes us sort of less people-like, and so forth. But it was very interesting to see how other people argued. And I realized that they argue differently from us. They don't argue from evidence. They argue from, source quoted or something. So it's a very different kind of evidence. So I thought, well, I can stick to scientific evidence, and that's the one thing I'll try to do. And so I ran afoul of the chair of the council, who basically mostly wrote the reports of the council, because I kept fighting over the wording. And I was actually not happy with the way some of the reports ended up reading with respect to the science, and decided I should say so. And with Janet Rowley, and in some cases with Michael Gazzaniga, we tried to make public statements.
You say publicly. What did you do? Write letters to the newspaper?
So we did two
or
things.
What?
Not to the newspaper. No, we wrote a letter to Science. And so we finally said, look, here's the final report. It's coming out under our names, but the science in it is still not correct. And showed the chair, and the chair was very upset and said, you have to withdraw this. And then another report was being prepared. And again, it ran into the same problem. And this one was on the status of stem cell science. And again, we thought it's not laying out where things are. So we wrote another letter and sent it again to the chair and said, look, we're sending this letter off to Public Library of Science as a letter. Again, because if the report comes out, we thought the record should be corrected about where the science is. And so then, after that, fairly shortly, I heard that I wasn't going to be renewed on the council. So, so that was the sequence of events. And as I say, one can only sort of backtrack about what led to the decision.
Liz, the list of scientific honors that you've received is extraordinary. beginning with the election to the Royal Society in 1992 and the U.S. Academy of Sciences in 1993, and a list of prizes and other honors that's really too long to begin to mention. What is your reaction to all of this notoriety, if you will?
Well, the first time I got a prize, it was very interesting because I felt more gratified than I thought I would. It was like, so, you know, or people accept what I do, or something like that. It was very interesting. Now, if I get an award, there's this sense it's important because people need to see that women scientists can get awards.
Yes, well, you actually led right into the next question I was going to ask you, which was about the problems that women have in science.
You know, for many years I just blundered ahead as though I was sort of gender-free, and that was, my reaction was very much, you know, an ostrich reaction, which is to do the science and just try to ignore anything else. Having my own child and own son certainly highlighted the realities of, you know, if you're somebody with a family and you're dealing with a family, I suddenly realized, you know, what an enormous challenge that was. And our son wasn't born until I actually was promoted to full professor. We just see the numbers telling the story, you know, plenty of women in graduate school, many women in postdoctoral fellowships, top institutions. Where are the women applicants for assistant professorships? Suddenly the fraction has dropped, at least at high echelon sort of institutions, like the one I'm in, the search committees I'm on. It's just appalling, the numbers who are applying of women versus men. And then it gets more and more extreme, and then when it comes to issues of power, as when I was chairing, that's the only time I've become in a situation where I felt it was disadvantageous to be not a male chair but a female chair. And I was the only one in the School of Medicine.
Do you mean that there were conversations that other people had that you were not
yes,
privy to?
Those things go on, and those things get worked out by various sorts of means that, you know, often involve the sort of networks and things like that that are not necessarily ones that I was necessarily able to go to, not least because I wanted to be home for dinner with our son. I think at the sort of scientific level, you know, in the scientific playing field, women play extremely well, right, and do very well. But I think it's the other aspects that are still very tough.
What do you tell your young female scientists now?
I think I try to encourage them to, you know, to recognize that they are really good, and if they want to do something, then they have the innate ability to do it. You, by the way, did that very thing for me as a postdoc, by the way.
So, a slightly different question. Your husband, John Sadat, is himself a prominent scientist who has done work on chromosome structure and also in microscopy. What's it like to have two eminent scientists in the same family?
It's been something where our arenas are sufficiently differentiated, but we can talk science to each other, and especially we can talk about, you know, all the things that go on in the life of scientists.
Are there things that you feel where you might have made a better decision, or things would have gone much better for you, or more comfortable, or more rewarding if you had done something different?
Where I've done something where I've kind of stepped out of what I think is what I would think is the right way to do it, that's usually when I've regretted doing it. And so, you know, the answer is that, you know, you should kind of listen to yourself, kind of, you know, I should be listening to what I feel is the right thing and not try and assume somebody else's idea of how you go about things.
Let me end this conversation then by just asking you very briefly, what aspect of your career has given you the most satisfaction?
Oh, I think the puzzle solving, when you're doing this science and you start to see that there's an answer and you think, I think I know how this works. And you know, just that excitement that you sometimes get when you do experiments, it's really, you know, I can tell that just makes me feel so thrilled when I have that kind of experience. And that can happen, you know, on any day at some level.
I think we are very much in accord on that issue.
Well, Joe, I think you very much had a big influence in letting me see the joy of that kind of aspect of science.
Well, thank you very much, Liz.
Thank you.